So, hello and welcome everybody to Eppendorf session here on LabRoot's virtual event series on cell and gene therapy. This session is titled Seed Train and Bioprocess Strategies for Suspension Cell Culture-Based Adeno-Associated Virus Production. And your webinar is going to last about 60 minutes, including a Q&A session. And it's presented to you by my two lovely colleagues, Ines Hartmann, application specialist here in Hamburg, and Stefan Schlößer from our bioprocess team in Jülich, who is a scientific communications manager. I will give you an introduction into both in about a minute. My name is Christian. I'm marketing manager here at Eppendorf, and I'll be a moderator today. And at the end of the presentation, I will also present you some of the solutions that might be helpful for your workflows before we just start the Q&A session at the end. Before we get started, I would like to all of you encourage you to ask questions to our experts. If you want to do so, you can do that at any time during the presentation. Just use the Ask a Question box on the left side of your screen, and we will answer as many questions as we have time for at the end of the presentation. If you have trouble to see or hear the presentation, just click on the support tab at the top right or report your problem by clicking on the Ask a Question box on the left. Now, let me do a little introduction of our speakers today, and I'm really excited and happy to have them here. Let's start with the ladies here, or the lady in this case. Ines Hartmann studied biology, and she joined Eppendorf already 17 years ago in 2018. She has been an application specialist here covering various products group, but really found her passion in supporting our R&D team and also customers with their applications regarding shakers worldwide. And if you don't find Ines in the lab here at Eppendorf, it's very likely that you will see her on the dance floor because she just loves to dance to all kinds of 1920s swing music. And that's also one of the reasons why she organizes an annual small swing festival here in Hamburg together with friends. Dr. Stefan Schlößer studied also biology, and after his PhD gained a lot of experience in microbiology, virology, and also cell biology. And he started 3 years ago here at Eppendorf as a scientific communications manager. And this is a way how he can live his passion to convey scientific knowledge to people. And here he really focuses on the very complex topic of bioprocessing. And in contrast to dealing with complex science all day, Stefan owns and expands a really impressive collection of retro video games. So the older ones of you may remember the Commodore 64, a Sega Mega Drive, or various Nintendo Game Boys. And as I said, I'm really happy to have those both of the speakers here today. And I'm really happy that they are going to share their knowledge with us. So I will stop talking now and hand it over. Ines and Stefan, stage is yours. Thank you.
Thank you, Christian, for this really, really lovely introduction. Also, warm welcome from my side. I'm happy that you join Ines and me today to our talk about seed trains and bioprocess strategies for suspension cell culture-based adeno-associated virus production. Let's first a quick look at our agenda today. We will start with a little introduction on how AAVs are used in gene therapy. Then we will have a look at the work from our application specialists, where they were producing AAVs in single-use bioreactors. During this part, Ines and me will give a little insights into strategies for seed trains. And after the conclusion, we will have a little look on the Eppendorf equipment that was used together with Christian. So without further ado, let's start with the introduction. So let's have a look at AAV. For those who are unaware, AAV stands for adeno-associated virus. It was first described in 1965 as a contaminant of rhesus monkey kidney cell cultures. It is a quite small virus, only 25 nanometers in diameter. It is non-enveloped and a member of the Parvoviridae family. It has an icosahedral capsid, meaning it is made up of little triangular structures, which you can see on the bottom left. And inside this capsid, it contains a single-stranded genome of about 4.7 kilobases. AAV alone is replication deficient. It lacks certain genes for full replication cycle. And this is why it needs a so-called helper virus to enable the full replication cycle. This is also why it's called adeno-associated virus, because the adenovirus is one of the helper virus, not the only one, but it is one that when co-infecting the cell where adeno-associated virus is inside, its enzymes can also help AAV to fulfill its replication cycle. AAV alone, however, is considered to be non-pathogenic, which makes it a very interesting target for gene delivery systems in disease treatment. And one of these approaches how AAV can be used to treat diseases, we will have a look at now. And this is the RPE65 mutation-associated retinal dystrophy. This is an inherent genetic disorder in the eye that can cause severe vision loss within the first year of life. And the reason for this is a mutation in the RPE65 gene that affects photoreceptors in the eye. To get a bit more into detail, RPE65 encodes the enzyme retinoid isomerohydrolase. And this enzyme regenerates chromophores that are important for light detection in photoreceptor cells. And if there's a biallelic loss of the RPE65 function, this can lead to impaired photoreceptor sensitivity and will ultimately lead to photoreceptor degeneration and loss of eyesight. And 8 years ago, in 2017, an amazing tool was introduced to treat this disease. It is called Luxturna and is the first FDA-approved AAV gene therapy. To just explain it quite simply, AAV is here engineered in a way that the viral genes are replaced by a functional RPE65 gene. And this makes basically a viral vector out of this AAV particle, which can then be injected into the eye. Here, the vector will enter the photoreceptor cell and deliver the healthy gene into the nucleus for expression. This means the patient suddenly has a functional RPE65 protein with an enzymatic activity to restore chromophores in the eyes. And this can then restore light detection and eyesight. Another property of AAV is that after its single-stranded DNA genome is converted to double-stranded DNA, it can persist in the nucleus as an episome, so little circular-shaped DNA structures, which ensures a long-term gene expression. So it's amazing what you can do with this little guy. However, the therapeutic approach with AAV also comes with some challenges. And one of them is the requirement for high doses for AAV treatment. If we look at Luxturna, and keep in mind this is only a treatment localized only to the eye, you already need 150 billion vector genomes, or VG for short, per eye. And if you go one step further and you want to have a systemic approach, clinical trials that are currently going on use doses of more than 100 trillion viral genomes per kilogram body weight. So these are numbers that are hard to satisfy with our traditional cell culturing methods. So what you would need would be a system that has the possibility to scale up to the production scale you need. It should provide stable culture and reproducible culture conditions. And this can be performed, for example, by precise control of the important process parameters. And if we look at this, bioreactors could be a possibility to tackle these challenges, as they cover a large variety from small scale to commercial manufacturing scale. They enable precise control of the necessary process parameters like temperature, pH, gassing, and so on. They also reduce the workload because they can automate certain working steps in process control. and they offer the possibility for adherent and suspension cell culture. So after this little introduction, let's now look at what our application specialists have done with this knowledge. And they were looking first at the general process of AAV production. And I want to go through here quickly. So usually if you want to produce AAV in cells, you have 3 plasmid types. First one is the plasmid PT, or transfer plasmid. This is usually the plasmid where you put your gene of interest in. So in our example, this would be where the functional RPE65 gene would go. Second plasmid is the PRC, or the rep-cap plasmid. This contains the AAV structural and packaging genes. So basically, it contains the genetic information for the capsid and how to package information like the gene of interest into this capsid. And as said, if you want to produce AAVs, you need to have a full replication cycle, and for this you usually need a helper virus. However, introducing another virus into your AAV production can be a safety risk, and therefore you use here also a plasmid that only contains the helper virus associated genes necessary for producing recombinant AAVs. And this is basically what you need to put in your cells and produce viral vectors. And our application specialists took this approach in a proof of principle study to produce AAV capsids in single-use bioreactors. And the difference here is that they used also this so-called helper-free AAV production system that I described, made of the plasmids, but they left out the plasmid PT. So the gene of interest was not included, as this was a proof of principle study. It was enough to only produce the viral capsids, which is the result of leaving out the PT plasmid. What did they use for this? They used a suspension-adapted HEC293 cell culture called XP293, which is from Thermo Fisher Scientific. And to produce the inoculum for the bioreactor culture, they did this in a seed train using shake flasks. And once the cell number was high enough, they inoculated the bioreactor, expanded the cells in there, transfected them in there, and produced the AAV capsids in there. And this is just a short overview of the seed train they were using. So as you can see, they started with thawing the initial cryo cell vial. It was put into a single shake flask and then incubated until the necessary cell density was there to split the culture and separate them to 3 new flasks. Then the process was repeated, and once the cells were dense enough, they were split again into more flasks, and this was done as long as the necessary cell number was reached to inoculate the bioreactor. So as you can see, the seed train is a simple concept, but it can take a lot of time depending how many cells you need, and the process itself bears risks to be avoided. So the question is now, what should we look out for during the seed train process? And this is where I hand over to my colleague Ines.
Thank you, Stefan. Oh yeah, let's get a little bit into the detail what is happening before the inoculum is going to the bioreactor. So before you go the other direction in whatever size of bioreactor you inoculate in, either a parallel bioreactor or in this case a Cyvario twin bioreactor. So we look here on the first part, so the thawing process and the shake flask cultivation. So the challenges whenever you prepare an inoculum is first it must be available in a sufficient density at the planned inoculation time. So you have to do proper planning, and a very typical bioreactor inoculum density is 0.2 to 1 million cells per milliliter, talking here about a traditional seed train process, not an intensified one. Also, you need the cells in a viable state to achieve a short lag phase in the bioreactor. Plus, of course, it should allow for productive AIV capsid production in this case. And also, it is sure, because especially shake flask, you handle them also in an open environment, in a shaker to a biosafety cabinet. So it must be free of contaminations. And also, it should be reproducible in the end, because you would do it more than one time. So the first thing, which is often not looked at so much, is the thawing process. And what we did in this application, or what is also our standard, to use not thawing in a water bath, but a programmable water-free thawing process. In this case, we used our thermomixer, which has exchangeable blocks. And we have like a smart block you can click on, so you can use also other blocks, but we have the smart block cryothaw, and this allows programmable thawing of not only one vial, but up to multiple vials. So if you have 5 vials in a row, you can, probably more is not handleable. For the later process, you can do it in parallel and prepare, for instance, your biosafety cabinet or your workspace. Here you see we have a lot of application and performance sheets that the viability and also it's not depicted here, but the morphology of the cells are the same as with the water bath thawing. We also did it with very sensitive stem cells, so just have a look if you scan here, or in the end Christian will also talk about the product. Next, that is quite a standard process, so we transferred it to pre-warmed medium. So to pre-warm it is always a recommendation, and also to avoid vigorous pipetting. So droplet-wise, you should add from your vial the cells into the next vessel. We always add a centrifugation step to remove the DMSO, and then we suspend it to check for short viable cell density and microscopic check, and seed it into a total small flask first, so 125 ml shake flask with, go to the next slide, with 20% fill volume. And the cultivation conditions are according to the protocol. The XP protocol is 37 degrees, 8% CO2, 125 RPM shaking, and we used a shaking diameter of 25 millimeters. So the expansion, we did 3 passages, as you see also on the picture here. What we did is to keep consistent seeding intervals, and also we kept consistent densities, so 0.4 million cells per milliliter, and in the end of the 3rd passage, we achieved more than 900 million cells in the 3rd passage. So the keynotes is, do before a growth curve of your cell and see where the logarithmic phase of growth is to ensure the right passaging phase. Also, in this case, we use baffled shake flask. This is not always a recommendation, so unbaffled are often the recommended one. Just in this experiment, we use baffles, which also work very well. And what we also care for is to minimize disturbances, so organize also in the incubator and reduce the steps outside the incubator to a very short time, and to have optimal conditions and the highest cell viability in the end. Yes, what we had also in the end, so the inoculum after 3 passages, we had more than 900 million cells, and we had like 2 inoculums because we had 2 bioreactors, 2 bioreactor vessels in this case, as the Salvario twin can handle 2 bioreactors with 1 controller. And we had 400 million cells, XB293F, in 200 ml with a cell viability of more than 95%. And we inoculated each BioBlue single blue bioreactor with starting density as well of 0.4 million cells per milliliter, and the final volume was 1 liter. What we did, or what is standard, we use these flask, which you can use also in unsterile environment. So with a transfer cap you can use, and here I think it's a standard method to weld it to the harvest line the line of the bottle with the harvest line of the bioreactor. And if you have small inoculums or just wanted samples on the left-hand side, do I have? Oh yes, I have an animation. Also, this allows for quick. The sampling caps are handy to allow aseptic and quick sampling. So to sum up, also, safeguard your inoculum train, so the time before the bioreactor. So of course, apply strict aseptic techniques, organize your workspace ahead, prefer water-free thawing to reduce contamination risks from water bath, and sample and transfer kits we find quite easy to handle, and it also minimizes the need to open the vessels. And also in between, also regularly disinfect surfaces of your shaking incubator or CO2 incubator shaker. And if it has a decontamination program, run it between batches to be secure. And with this part, we go back to the bioreactor, and I hand over to my colleague Stefan again.
Yeah, thank you, Ines, for this nice overview of seed trains. I have one additional point to make here. Ines was talking mainly about, we call it the traditional seed train, which is done in shake flask, which is a proven and established approach. But she was also mentioning quickly seed train intensification. And this is an alternative way. For example, if you're working with large bioreactor volumes, because let's say we think of a real big bioreactor with 3,000 liters of volume. This is actually what comes from the literature. It can take several weeks for a seed train to be established in a traditional way, a shake flask, for example, or bags. And this means you need to constantly transfer your culture many times. You need to change the medium many times. And this not only increases the maintenance workload, it also increases the risk for contaminating your inoculum culture. This is why a bit over 20 years ago, it was suggested to intensify seed trains as an alternative approach to the traditional one. It was introduced in 2002. It was suggested the first time, and over the last decades, it was further improved, and is now also a way to get your inoculum ready. And let's see what seed train intensification looks like. On the top here, you have first the traditional seed train using shake flasks, where we start usually with the started cryovial cell stock, which is followed by multiple expansion step in flask, as we described. And as Ines said, usually you end up with inoculum densities for your bioreactor between 0.2 and 1.0 times 10 to the 6 cells per milliliter. This is also the range which we will talk about later in our study. However, for larger volumes, you can also do the alternative approach of intensification, which means you start your seed train already with a very high density cryo cell stock. This is usually then prepared beforehand, and you can then store it either in a vial or in a bag, depending. And these starting cultures can contain tens to hundreds of million cells per milliliter, compared to usually a million in a usual cryo vial, for example. And of course, because you start with a higher cell density, you can reduce the total number of steps in your seed train. So you take basically your frozen bag or vial, thawed, and then you can, for example, even put it immediately in a bioreactor culture, where you then grow the cells to get your inoculum ready for the next big bioreactor, where you, for example, produce your product in. And this would then be, for example, a reduction towards a two-step seed train. So if you're working with higher bioreactor volumes, this is definitely an option to look out for. Okay, but now let's go back to our application note here. Our application specialist, as said, worked with a traditional seed train where they used shake flasks to grow the inoculum in one of our New Brunswick S41i CO2 incubator shakers. And once the cell density was high enough, it was used to inoculate 2 BioBlue 1C single-use bioreactors. And if you look at the table at the bottom, we have the process parameters, as Ines said already. The final working volume was 1 liter. It was done in batch culture, which means the culture was undisturbed during the bioprocess and was only disturbed when the process was ended. Of course, the cells were incubated at 37 degrees. The agitation of the impeller was at a speed of 155 RPM or 0.4 meters per second of tip speed. The inoculation density in the bioreactor in the end was 0.4 times 10 to the 6 cells per milliliter. And the DO set point, so it means the DO was kept constant at 40%. Also, guessing cascade was implemented for this to ensure this. And the pH was also maintained at 7.0 during the process. Controlled were all these parameters by the Savario twin bioreactor control system, which you can see on the right side at the bottom. And this is a bioreactor control system where you can connect 2 bioreactors at the same time, and you control them individually or in parallel. And in our case, we choose to use them in parallel. In that case, we also had a repetition of our experiment right out of the bed. And just to go through what we did in terms of the workflow. So our application specialists, as mentioned before, they used basically an AAV helper-free production system without the plasmid PT, so without the gene of interest. And so they only used the PRC and the P helper plasmids in order to produce basically empty AAV capsids, which is suitable for our proof of concept study. To get the plasmids into the cells, the cell were then expanded in the BioBlue 1C single-use bioreactors under control of the Savario twin. And in the next step, they transfected the cell with a transfection reagent called Factovir from Polyplus. And afterwards, the cell were able to produce bioreactor capsids. These are basically the results and how it was done in detail. We can see that the whole experiment timeframe was 7 days. And 3 days after inoculation of the bioreactor, when the cells reached about 3 million cells per milliliter, the transfection was done. For this, the plasmids, so the PRC and the P helper plasmids, were mixed at a molar ratio of 2:1, together with XP293F expression medium. And hereafter, they were mixed with the transfection reagents, so Factovir, in a ratio of 1:1. And then after incubating it for 30 minutes at room temperature, the sole transfection mix was added to the bioreactor. And then the process was further observed. So every day before and every day after the transfection, cell samples were taken and cell density, viability, and the metabolite concentrations were measured. So let's have a look at the results of this. What we can see is that we had efficient cell growth with a peak cell density of 7 million cells per milliliter at the last day. Before the transfection was done, we could see that the viability was larger than 90%. However, after the transfection, so from day 3 to day 7, we could see that the growth rate as well as the viability was reduced. This is something you expect with viral transfections because the cell is afterwards busy producing the capsids, which puts a lot of burden on the cell and thereby reducing the cell fitness. From unpublished data from our application team, I can also say that if you don't transfect the cell and you let the process run with basically completely untreated cells, you can reach up to 13 million cells per milliliter in the bioreactor. What we can also see from the top figure is that the processes were highly reproducible. So the two bioreactors share a very similar profile in terms of cell density and viability. And if we now look at the bottom figure, we can see that glucose is basically consumed constantly by the cells, which is expected when they grow. And at the same time, growth inhibiting metabolites like lactate and ammonia were maintained below toxic levels for the cell. So after we see now how our cells are growing, we now go to the next step, which is isolation and quantification of the AAV2 capsids. The cells were harvested, as well as the medium was harvested 72 hours post-transfection. And then the AAV capsid were isolated with the AAV Pro purification kit from Takara Bio. This was done for the supernatant and the cell pellet. Usually the supernatant is the more logical choice because this is where you get your viral vectors from in the end, but we wanted also to see how many viral vector capsids we can get out of the cell pellet. And after the isolation step, the capsids were then quantified by the AAV2 Express ELISA kit from Progen, and the results can be seen here. Just for explanation, on the left side, you see the results for bioreactor 1. On the right side, you see the results for the second bioreactor. The darker colored columns are the results for cell pellet and the lighter ones for the supernatant. We can see here that we got robust AAV2 capsid titers of up to 10 to the 11 capsids per milliliter in the supernatant and 10 to the 12 capsids per milliliter in the cell pellet. You can also see here, as before, for the cell density and the viability, that there's an excellent reproducibility between the two bioreactors, thanks to the precise control of the Saverio-Twin bioreactor control system. We have basically very, very similar numbers in virus titers. And with this, we can already come to the conclusion. What was demonstrated here is a Stirling bioreactor-based process for producing AAV2 capsids in suspension cells. We could see that the Saverio-Twin bioreactor system, in combination with the BioBlue 1C single-use bioreactors, as well as a good seed train before, of course, resulted in efficient cell growth and AAV capsid titers from 10 to the 11 to 10 to the 12 capsids per milliliter. We could also see that the process were very reproducible, thanks to the precise parameter control. And one thing that is to mention is that the study here describes an efficient viral vector production platform approach that can be used for gene therapy research and development, because the equipment that was used here, so the Saverio-Twin and the BioBlues, are mainly usable for research and development purposes. However, Eppendorf is also offering bioprocess solutions if you want to go into clinical and commercial manufacturing. And we will hear more about this in the coming slides together with Christian. And to you, I hand over now for the overview of the Eppendorf equipment.
Yes, thank you very much, Stefan. Thank you also for your nice presentation, also to Ines. I could already see during your presentation that there were some questions coming in, and we will talk about this in a second. But before we start the Q&A session, I promise you to give you a little insight into product solutions from Eppendorf that might be helpful for your daily work. And yeah, let's start with that. The first product that you probably are familiar with is our Thermomixer C, because it's a very popular product from Eppendorf. It's available with a lot of smart blocks that you can attach to the device. So it's very easy. As you can see on the left side, you just push the little lever there, and you can exchange the blocks with different vessel types. and also the machine recognizes which kind of block you have attached to the thermomixer. And what might be most important for you here is that we also have a smart block of cryothaw. So this is for water-free and maybe more importantly, standardized cell thawing that you can do with your cryo vials. So no hand warming or water bath warming. So if you already have a machine, especially that might be interesting, because you just need to buy the cryo thaw block and attach it to your thermomixer. And the other smart block that might be interesting for you here is a smart block for 50 ml conicals that you can use for warming your supplement aliquots, for example, or media aliquots. So if you want to learn more about this, you can go to the page with the link below, eppendorf.link/thermomixer-C. The next product I would like to highlight, and Ines already talked about this, is our CO2 shaker, New Brunswick S41i, which comes with a static shell for adherent cell cultivation. And underneath you find a shaker platform. So this is our smaller module, which is stackable up to 2-fold. And this really supports reproducible inoculum preparation because it offers you effective protection from medium evaporation due to the water trace inside. Comes with a very low CO2 consumption, which you can see in this little graph here in the middle. And more importantly, when it comes to contamination prevention, as Ines mentioned before already, is a safe and easy reset in between your projects, in between your batches. So it comes with an integrated 120-degree disinfection. It's actually the only device on the market with this feature. And it also comes with a seamless stainless steel chamber, which you can see here in the middle. So it's very easy to clean, very fast. So you don't need to invest much time to clean your device because of the fanless device. Regarding the static shelf, you can also include a second one if you want to use it more like a small CO2 incubator. That's possible as well. If you want to get more information, you can check out our CO2 incubator or shaker session on the Eppendorf website. Stefan already talked about our equipment for R&D. This is also the equipment that has been used in the study that Stefan mentioned. So it's our Saverio twin bioreactor control system, which you can see on the left, with a working volume of 60 ml to up to 40 liters, which comes with a very intuitive user interface. And as Stefan also mentioned, it can control up to 2 bioreactors from this one controller. If you want to learn more, choose eppendorf.link/Saverio. And together with that, you can use our BioBlue-C single-use bioreactors for cell culture. You can see a little selection here. Those are scalable, available in different formats from 100 milliliter to up to 40 liters here. They really reduce cross-contamination risk or contamination risk in general, and they can reduce turnaround times and cleaning efforts. And I've seen there are also some questions regarding this. We will come to that in a second. However, if you have questions or if you want more information, you can go to eppendorf.link/BioBlue-C. And last but not least, before we start the Q&A session, we have also equipment for clinical and commercial manufacturing. So we have validation guides that support you in qualifying your equipment, the BioBlue H&Q single-use bioreactors, and also the BioFlow 320, which you can see here on the right side, the bioprocess controller, which are for clinical and commercial manufacturing, as Stefan already pointed out. The software here, the Dustware Control Plus SCADA software, is for use in clinical and commercial manufacturing and is compatible with 21 CFR Part 11 and the Udrelex Volume 4 Annex 11. If you want to learn more, go to eppendorf.link/commercial-bioprocessing. And with this, let's jump right into the Q&A session. So if you have any questions for our experts, please put them into the Ask a Question box on the left side of the screen. I will make sure that we select the most or the one question that might be most relevant for most of you. And let me open a little box here to check the questions that we have. And we start with one that's probably one for Ines. The question is, would you recommend using baffled or unbaffled shake flasks in general? So you already touched about this topic a little bit, but apparently there are some more detailed questions on this. Ines.
Yeah, I mean, in general, when it's about comparing cultures, the non-baffled is more promoted, I would say, so from literature and so on, because the baffles, if it's in the bottom, it can create, or it creates, of course, more turbulences in the shake flask. But on the other hand, in this study, we used baffled flasks, and we have also good experience with these. So I think, especially in AAV production, both, of course, vented, non-baffled, and baffled are used. So I would start with non-baffled and just try. So it's a little bit more popular, the non-baffled, but if, for instance, if you work with bacteria and yeast, which grow very quickly and consume a lot of oxygen, to use baffled flask is very, very common, very handy, especially if you want to increase the volume, so you can get more oxygen in the culture broth, but for mammalian cells, it's good to start with non-baffled. Of course, on the other hand, there are alternative flask types like special alternatives like optimum growth and so on. They have special baffles. We have also good results with these flasks. So both are fine to use. Pros and cons. Give it a try. Both.
Yeah, but you would recommend to use unbaffled because of the shear stress.
Yeah.
That's
yeah, it's more about
okay.
consistency. I don't think the shear is such a problem. When you have very shear sensitive cells, I would go for non-baffled.
Okay, so start with the unbaffled first. All right, thank you very much, Ines. The next one I can see is for Stefan. Good question. Why were single-use bioreactors used in this study and not glass bioreactors?
I mean, both have their valid use, I would say. However, if we look on the approach here, which is AAV production, and if we now think about that we are also using our equipment for clinical and commercial manufacturing, for example, there are quite some rules that you need to basically fulfill, and especially about how clean your bioreactor needs to be. And the advantage for the single use is that you have a reduced cleaning effort because you basically use it once, and then you don't need to put the effort in the cleaning. For a glass that is reusable, which can also be in certain cases an advantage, but you need to have a cleaning procedure worked out, which can be a lot of work and can also come with insecurities depending on what is needed for your bioreactor. So in general, the single use, this is also why they are gaining track in the field of cell and gene therapy, for example, in the recent years. The handling here is just easier. Plus, as we said, if you have a long process, there's also less contamination risk. So
yeah, you have to guarantee that they are sterile, right, during delivery.
Exactly. You can just run your process with a more calmer mind, I would say.
All right, thank you, Stefan. There's also a bioreactor-related question. The question is, you used suspension cells here, but is it also possible to produce viral vectors from adherent cells in bioreactors? Stefan.
Yes, this is definitely possible. And you can see this, that the HEK cells that were used, they are suspension adapted, but the original HEK cells, I was working with them also in the lab some years ago. they are usually adherent cells, so it means they need a surface to attach to. And if you want to realize this in a bioreactor, you can basically use certain scaffolds. For example, you have something that's called a microcarrier, which are little beads, for example, made from polystyrene, but there are also different materials used, which you can add to your bioreactor, where the cells then can attach to, and then they can grow. I would say for cells that, like HEC293 cells that need an attachment surface, this would be an option. And by the way, using these makes also scale up easier, because you can just add more of these microcarriers, for example, to your culture, and then the cells will have new surfaces to attach to. And you don't need to like change the whole bioreactor for your next scale up step. So this is one option, for example.
And detachment from those microcarriers is also done like an adherent cell culture with trypsin, or how does that work?
This is usually the way. There are different variants of it, but this is usually the standard that is done at the moment. For producing bioreactors, it is not so critical, actually, the detachment step, because you usually take your bioreactors out of the supernatant.
Yes,
so for this, it's no problem, but yeah, usually it's the typical detachment methods, even though people are working on alternatives for this as well.
Thank you, Stefan. Next question goes to Ines regarding shake flasks, and the question is, what fill volume in shake flasks would you recommend? Yes, a very common question.
We usually look at the protocols and also what the flask suppliers recommend, because there are actually many different types of flasks, so it's a little bit depending with which flask you work. And also it's good here to try out different fill volumes, maybe in the beginning when you set up your culture, but in general between we use here 20% up to 40% is for mammalian culture a good rule of thumb.
But I've seen also newer flask with higher fill volumes.
I mean, these for optimum growth flasks, as I said, they have different designs, so they have a more steep neck. But you have to use them also with a little bit of higher RPNs agitation, and you can go also to higher fills. So 40% is, I think, more or less the maximum.
Perfect. Thank you very much, Ines. We have two more questions here, so if there are no further questions. The next one is regarding thawing. The question is more product related, I guess. Is your thawing block for automated cell thawing limited to certain vials? Is this related to the shape of the vials or the volume? Ines, it's probably
you can use, it's probably me. You can use 1.8 to 2 ml, the standard vials. It is optimized, or at least our protocols are used with 1 ml fill. So if you use higher fill volumes, you can of course adjust the program. So in the beginning anyhow, you need to test the program and you can adjust the steps, but we optimize it for 1 ml fill and standard 1.8 to 2 ml vials, cryovials. So if you have any uncertainty, reach out to us or to your local Eppendorf contact. So we are happy to help you.
Yeah, I think we also got this information available on the website. All right, let's go to the last question that I can see here, and this is also bioprocess related. And the question is, are there other options to monitor your process than directly on the bioprocess controller in the lab? Stefan.
Actually, there are. I didn't mention this directly, but of course, you can always go into your lab, check how the process is running on the screen of your bioprocess controller. But Eppendorf also recently launched a cloud-based solution for this, which is called BioInsight Cloud. This is basically a little program which is also included in the dustware control from version 6 upwards. So we were talking quickly about dustware control. It's part of that, and it enables you basically to monitor your bioprocess values on the go. You can exit it from, for example, a browser on your phone. You can also check it while you're on the road. You can share the access with different people. So it makes it a lot of easier. Let's say you're on a conference and you just want to see if your process is running well. You can quickly check it. It also enables you comparing your actual run with past runs, or comparing past runs with each other. And it also offers some AI-supported tools for more further or detailed analysis. Yeah, so this would be one of the recommendations if you want to watch what your cells are doing when you're not in a lab.
All right, thank you, Stefan. Thank you, Ines, also for your answers. I cannot see any more questions, so therefore I guess we are at the end of this presentation. Thank you, Ines and Stefan. Do you have any final comments for our audience here?
Then I start. So thank you very much for attending today's session. I hope you enjoyed it. I hope you learned something. And yeah, if you need anything from us, just get in contact with us and let's bioprocess together, I would say.
And let's shape together. Also.
Yep. Yes.
Thanks also from my side. If you want to hear about any topics, please post it. You can send it, I think, also after this session to us in this chat.
Yep.
And would be great because we plan also, of course, future webinars. If you're interested in specific topics, then let us know.
Yeah.
Thanks.
Yeah. Before we go, thank you, Stefan and Ines, for your brilliant presentation, for taking the time to answer the questions. Before we go, I would like to thank all of you out there for joining us today and also for your questions. If you have submitted questions, you can also submit questions later. They will be addressed by our speakers via the contact information you have provided at the time of your registration. We would also like to thank LabRoots and their brilliant support today. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay, and we encourage you all to share that email with your colleagues who may have missed this live event today. And until we see us next time with any webinars from Eppendorf, we say goodbye from Hamburg and from Jülich. Bye, everyone.
Goodbye.